Hello friends, good afternoon and welcome to EduSat Live Lectures. Dear friends, today in Zoology, we will be talking about Developmental Biology and Introduction. To discuss this topic, we have with us our subject expert, Dr. Charu Dogra Rawat. Dr. Rawat is Assistant Professor in Department of Zoology in Ramjas College, University of Delhi. Without further ado, I would like to welcome ma'am to our studios and request her to start the lecture. Welcome ma'am.
Thank you. So today in this particular session, I'll be introducing the subject developmental biology to you. So developmental biology, what does it encompass? What do we study in it, or how do we study in it? So looking at different aspects that this subject covers, I'll be talking about today, and then in the successive series, we will be taking on each topic in detail. So developmental biology, there is another term which is very prevalent, which is called as embryology. So earlier, in fact, we used to study embryology, and now we have shifted to study developmental biology as such. Embryology is a slow process of progressive changes by which a multicellular is formed, is termed basically the development. So developmental biology looked at these progressive changes by which a multicellular organism is formed. The development begins with a single cell, the fertilized egg or a zygote that divides mitotically to produce all the cells of the body. The study of animal development has been traditionally called embryology from the phase of an organism that exists between fertilization and birth. So the onset of developmental biology or embryology is from the point where the two gametes fuse together to form a single cell called as the zygote. Now, how this zygote develops into a multicellular organism is what is encompassed in this developmental biology study. But initially we used to call it embryology because we used to study only the processes involved from the fertilization till the birth. But the development does not cease at the time of birth. The development does not stop or even at the adulthood. The replacement of skin cells, the generation of new blood cells, the regeneration of severed parts by some animals, and metamorphosis where a form which is called as the larva transforms into the adult form. That metamorphosis processes, these are all the evidences of sustained development. So development just doesn't stop when an organism is born, but it continues and it's sustainable and it involves all these different kind of processes. So developmental biology is a more appropriate term, and it is that discipline that studies the embryonic as well as other developmental processes. So basically, the developmental biology involves embryology as well as the other processes which continues after the birth of an organism, such as the growth, such as aging, which is when the growth deteriorates, the process such as regeneration in some animals, the process such as metamorphosis as we talked about. So all these is encompassed and included along with embryology, and that forms the developmental biology as a discipline. Now, developmental biology, what do we study in it? So, developmental biology primarily asks two questions, or basically there are two objectives of study developmental biology. And these two objectives are that how cellular diversity and order within each generation is generated. That is, how does a fertilized egg give rise to an adult body? So, we just saw that, the development starts from a single cell which is called as a zygote. So when the adult is formed from this particular single cell, how does that happen? That is the objective of studying developmental biology. Another objective is to study how this continuity of life is maintained from one generation to another. That is, how does the adult body produces yet another body? The objective of reproduction, the objective of environmental influences. So all these two major objectives of developmental biology generally asks a series of questions, which is the question of differentiation, the question of morphogenesis, the question of growth, the question of reproduction, the question of evolution, and the question of environmental integration. So let's look deeper into what these questions are, what constitutes these questions, and therefore what primarily constitutes the studying of the developmental biology. The question of differentiation is basically the generation of cellular diversity. Differentiation is a phenomena where a single cell tend to get form different kinds of cells. A single cell, which is a fertilized egg, give rise to hundreds of cell types: the muscle cells, the epidermal cells, the neural cells, the lens cells, the fat cells, and so on. So how can the fertilized egg generate so many different cell types? Now we know that every cell perhaps have the same genetic organization. Many of them contain the same genetic information they are carrying. So this genetic instructions, how they are expressed, or in what way they are expressed to generate different cell types. So this is the question of differentiation, which is a primary objective of studying developmental biology. The next question is the question of morphogenesis. The different cells that are formed, they are not randomly distributed. They are organized into intricate tissues and organs. Cells divide, they migrate, they die, the tissues fold and separate. The organs are always formed and arranged in an ordered way. The fingers are always on the tips of the hand. They do not originate anywhere else. So what create this ordered form? The creation of ordered form is termed as morphogenesis, and developmental biology again include this primary question that one single cell is giving rise to multicellular organism, but in that multicellular organism everything is organized and ordered in a particular manner. So how can cells form these ordered structures? The next question the developmental biology asks is the question of growth, which basically means that how do our cells know where to stop dividing? If the cells of your arm was supposed to divide just one more division, your arm has been really long, and probably you can pick things without even bending. So the arms are just the right size. Then both the arms, which are on both the sides, on the sides of the body, they are generally of the same size. Then how does this happen? How is the cell division regulated? This tightly regulated. How does the cell know that now it has to divide? It has to divide up to this number and then it has to cease dividing. So how does this growth occurs, and where does the cell know that okay, the growth has to stop? So this is again a primary question asked by the developmental biologist. The next is the question of reproduction. There are certain specialized cells inside the body which are called as the gametes, the sperm and the ovum. So the gametes are specialized cells that can transmit the instructions for making an organism from one generation to another. How are these cells set apart, and what are the instructions in the nucleus and cytoplasm that allow them to function in this particular way? So how they are separated from all the other cells, and they know that they have to fuse together and give rise to the next individual. So they are carrying that information. So this question of reproduction is again asked by developmental biologists and studied. The next question is the question of evolution. The inherited changes in the development constitutes evolution. How do these changes in development create new body forms? Now from generation to generation, we know that there is a lineage which is forming. There were ancestors, there are different progeny. So how does the progeny which is formed is different from the parent that is there? How this question of which heritable changes are possible given the constraints imposed by the necessity of the organism to survive in that particular environment. So how does it adapt? How does the changes occur? What genes are expressed, or what processes are there that constitutes that development leads to the evolution or the giving rise of the new organisms which are better suited to the environment. So how does this evolution occurs is again developmentally related question, and therefore developmental biology needs to study that. The next question is the question of environmental integration. Now the development is always influenced by the cues of the environment that surrounds the embryo or larvae. How is the development of an organism integrated into the larger context of its habitat? And what properties enable certain chemicals to alter the development? So, the development is not occurring in isolation. Wherever the larvae is there or wherever the embryo is there, there is a surrounding, there is an environment in which it is lying, and that environment influences the development. So, there are certain signals, there are certain environmental cues by which the development can proceed in one way or the other. So, what are these environmental cues that cell takes up, and how does the cell takes up these cues and develop in altered ways? So, again, developmental biologists need to study and seek answer to this question, the question of environmental integration. So, the question of differentiation, the question of morphogenesis, the question of growth, the question of reproduction, the question of evolution, and the question of environmental integration. All these 6 questions constitutes the main objectives of studying developmental biology, or main objectives, what are the basic objectives of developmental biology, that how does a single cell give rise to a multicellular adult, and that multicellular adult is better suited to the environment and survives and thrives. So, these are the primarily questions that developmental biology asks. Now, what are the approaches to study developmental biology? Primarily, there have been 3 major approaches: the anatomical approaches, the experimental approaches, and the genetic approaches. So, in this part of introduction, we will look majorly at the anatomical approaches. How does an organism look like? So, the anatomy of the organisms, they generally study the anatomy of the organism to understand about the development, how a single cell is converting into a multicellular organism, and they are doing so by observing the anatomical differences between the embryos of different species, in fact between the differences between the embryo and the adult, and talking about all these aspects. They do it in the anatomical approaches, and in fact they include the evolutionary aspect also. The experimental embryology approach is usually when the experiments are conducted, various tissues are removed, or there is genetic manipulation that occurs. So, various experiments, when they are done on the embryos inside a lab to study about the development of that particular embryo, then it constitutes the experimental approach. Genetic approaches, because now everything is regulated by the genes. All the proteins that are formed, all the development. So, there is a genetic core to the developmental studies. And genetic approaches is when we go to the molecular level, we look at what genes are expressed, how the transcription of certain genes are regulated in certain developmental environments, and we look at the molecular level, the DNA, the gene level, the protein level, how are they expressed. That constitute the genetic approach. So, all in all the 3 approaches are come together and tell us more about the development of an organism, or helps us to study the developmental biology. So, in this particular lecture, we will talk majorly about the anatomical approaches. What parts of the embryo form the heart? How do the cells position themselves appropriately? For example, the ones that form the retina distance themselves from one that forms the lens. How are the tissues in different organisms related? For example, the tissues that form the bird wing to that of fish fin or human hand. What organs get affected by mutations in the genes? And are there any mathematical equations that relate the growth of different organs within the body? So, anatomical approach tends to answer these questions, that what part is forming, which tissue is giving rise to which organ, how the cells are are undergoing this morphogenesis, this ordered form, how are these cells doing that, how are the tissues different in different organisms. So, they do have a relationship between different organisms, they do look at that aspect also. Then they do look at the evolutionary biology also, that how the things are evolving to give rise to a particularly in the case of development, how the development is leading to the evolution. Or are there any mathematical equations that can be derived, and there can be certain laws that the development is taking place in this manner. So basically the comparative embryology, the evolutionary embryology, the teratology or the medical embryology, and the mathematical modeling. All these things need to come together and constitutes the anatomical approach to study the developmental biology. Aristotle was the first person, who talked about. So let's before talking about what are we going to study in developmental biology, let's look at how the developmental biology came into existence. What does this thought of studying the the development started from, and then how this slowly revealed different aspects of studying developmental biology. So it dates back to the era of Aristotle. He was the one who noted different ways the animals are born, oviparity where the eggs are laid, viviparity where the young ones are laid, and ovoviviparity where the eggs are laid inside the body and the young ones come out of it. So he noted that there are different ways the animals are born. And he also identified two major cell division patterns, which we will talk about in detail when we talk about the cell division after a single cell zygote is formed, which are called as the cleavage. So the holoblastic and the meroblastic patterns of cleavage. Holoblastic when a cell completely divides, and meroblastic when the cell has a cell-free cytoplasm region or an area where the divisions take place, whereas the rest of the region, which generally includes the yolk, does not get divided. So holoblastic and meroblastic patterns of cleavage was the first noted by Aristotle. Then he was the basically first to figure out, first to figure out the different types of blastulae which was there. So he was the one who identified that different kind of growth patterns are also there, and then he was the one who further posed this thing that okay, this is how it occurs. Then came William Harvey. He talked about all animals including mammals, they originate from egg. So he was the one who first showed that all animals including the mammals, they are all originating from the egg itself. He was the first to see the blastoderm of the chick embryo. So actual development of the chick embryo, where there's a small region on the egg where the divisions take place. The chick embryo undergoes meroblastic cleavage, and the region of the egg where the division takes place is termed as the blastoderm. So he was the first person to observe this blastoderm, that the divisions are actually taking place. He also noticed the island of the blood cells from before the heart comes from. So where from before before the heart comes, the blood islands are formed, and he also observed that. So William Harvey was the one who actually studied the chick development in detail in those times. Then came Marcello Malpighi. He was the first one to publish the microscopic account of the chick development. For the first time, the neural groove, the muscle forming somites, and the first circulation of arteries and veins to and from the yolk were identified. So by this time they knew that they understood that how the development, they were at least observing anatomy, that okay, from where tissue, what region is forming. They were observing, they could observe the chick development, the first microscopic views. They could see how the blood islands are formed. They could see how the neural tissue is originating, the muscle tissue is originating. So basically observing the development was the major thing. So anatomically they could identify the adult structures, and they could see that how they are deriving from the embryonic stages. So there came an era where there were two schools of theories, which was basically the epigenesis and the preformation. So epigenesis is basically the organs of the embryo are formed de novo from scratch at each generation. So there were there was a division amongst scientists, and one school was propagating the fact that the embryo there are resources which are present, like when they observed the chick development, particularly Aristotle and Aristotle and Harvey were the one, and K.F. Wolf, who propagated this epigenesis idea that everything is starting from the scratch. However, there were another school of thought which said that the organisms are already formed, miniature organisms are already formed, usually in the egg, but sometimes in sperm also, and all the development is basically only the growth that occurs. So they are already formed, but what they do is the cells keep on dividing and giving rise to, they keep on growing. So there is where the organs are already present in the miniature forms, and during development all it does is to grow into the adult form which is there. And it was supported by Malpighi, Albrecht von Haller, Charles Bonnet, René Descartes. So all these scientists, they propagated this pre-formation theory. And in fact, the religious, and the philosophical beliefs at those time also pointed towards the pre-formation theory. And epigenesis was considered as, it cannot happen, because they know that, everything is, there is somebody who is creating an individual, and that individual is just growing during the development. So then came the era of Immanuel and Blumenbach, who basically, Immanuel Kant and J.F. Blumenbach, who reconciled these epigenesis and pre-formation theory. They postulated a mechanical goal-directed force, which is called as Bildungstrieb, which is basically a developmental force. So they said that, there is a mechanical, there is a purpose for the development to occur. So development is occurring because there is a developmental force which is pushing it for the organism to form. The force was inherited through the germ cells, and development could proceed through a predetermined force inherent in the matter of the embryo. So it was a reconciliation between epigenesis and preformation, which says that the structure is not preformed, but the instructions are already present. So when the organism is formed from its parents, then this information of formation is passed on. So information is inherited, it is predetermined, this developmental force is already present, but then development takes place in an epigenetic manner. So there is a force which is driving force, or the developmental force, or the instruction and information to develop is already present, that is preformed, not the structures. So the information and instruction is already there, but what develops is basically from de novo. So the instruction, so that was a reconciliation between epigenesis and preformation, and they also pointed out that this developmental force was susceptible to change. So depending upon the environment in which this force is working, it's not a constant force, but it is basically can change over time. So epigenetic development is directed by preformed instructions. So which is the most accepted thought, during the modern days, that okay, the information and the instruction is already stored in the genome, and that information tells, or that act as the developmental force for the development to takes place, but development of structures etc. take place from de novo. So single cell give rise to the multicellular adult, and tissues and organs are formed. So instruction is preformed, but the development is epigenetic. So that was an important aspect when it came, the Immanuel Kant and Blumenbach basically reconciled the epigenesis and preformation theory. Then came Christian Pander, Heinrich Rathke and Carl Ernst von Baer. Christian Pander discovered the 3 germ layers. So he talked about the ectoderm, endoderm and mesoderm, the primary germ layers which are present. He discovered the tissue interaction. So another phenomena which he discovered was the phenomena of induction. The germ layers do not give rise to organs independently. So it works as a whole unit. The development is not, isolated regions do not develop independently. There is always an induction which is playing on, and they lead to the specification of different organs. Then Heinrich Rathke, he observed the development of frogs, salamanders, fish, turtles, birds and mammals, and emphasized the similarities in the development of all these vertebrate groups. So now talking not just the development of one individual, he observed the development in various species from different phyla, and then he observed that there is a similarity in the development that is there. So it was observed by Rathke, and he was also the one who described for the first time the vertebrate pharyngeal pouches, which become the gill apparatus of fish, but in mammals become the jaws and the ears. So he observed these pharyngeal pouches, and in fact one of the structure is termed after him, called as the Rathke pouch, which is basically, he observed that these embryonic structures are basically similar in all the organisms. Now on the basis of these observations, he also described the formation of vertebrate skull and the origin of the reproductive, excretory and respiratory systems. So these are some of the important contributions of certain embryologists during those times. Now his observations was basically put down by Carl Ernst von Baer into laws, and he himself also postulated certain generalizations which became the laws of the developmental biology, that how the development is occurring. Carl Ernst von Baer was the one who discovered the notochord. He was the one to discover the mammalian egg. He was the one who also showed that the mammalian development also takes place from an egg, and he in fact could observe it. And the 4 generalizations he derived out of his observations are termed as the von Baer laws. And these laws are that the general features of a large group of animals appear earlier in development than do the specialized features of a smaller group. So, specialization always occur at a later stage. So, initially if you observe Darwin will develop all these laws and talk about the embryonic similarities and evolution therefore, but he was the one who gave this generalization that the general features, the common features which are required for all the species would be similar, and they will develop early in the development, and later on the specialization will occur and the specialized tissues or structures will be formed. The less general characters develop from the more general until finally the most specialized appear. So first thing is that the specialized features always occur in a smaller group. So in a larger group there are general features, and these smaller group diversions, divisions will take place because the more specialized structures will appear. These more specialized structures will appear from the less general characters which have originated earlier in the development. So specialization comes later in the development. Initially there is a general character which is there. Then the embryo of a given species, instead of passing through the adult stage of lower animals, departs more and more from them. So it's not that that the embryo of a given species will look like the adult of a, it's not a hierarchy, it's not a linear development that is taken place. The embryo of various species will have a general characters, but as they grow and become adult, they keep on diverging from each another because the specialized characters appear and they become the characteristic of that particular species. So the embryos but it's not like that the lower animal adult will be the embryo of a higher animal. It doesn't work in that manner. It's the generalization was basically that the embryo of an higher animal will, as they grow, will as the structures will specialize, they are going to become diversified and very distinct from the other animals which are there. Therefore, the early embryo of a higher animal is never like a lower animal, but like its early embryo. So there is a similarity between the embryos which is there. But as they grow and form the adult by the process of development, they tend to diversify and diverge from each other. So the lower animal is not the embryo of a higher animal. There is not that kind of a linear development that is taking place. So these were the generalization which came to be known as the developmental laws, certain kind of points that were observed and that was generally taken back. So these were the Van Baer laws which were there. And based upon this, then the cell lineages started. So cell lineages and the cellular basis of morphogenesis. So with cell being demonstrated the basis of anatomy and physiology, the embryologists started tracing the cell lineages, following individual cells to see what they become. By tracing cell lineages, the fate maps could be drawn. Now a fate map is a diagram that map the larval or adult structure onto the region of the embryo from which it arose. With the information these fate maps provide, they are considered the basis of experimental embryology. So when the cellular basis of development occurred, that the cell is basically giving rise to different types of cells, and they started following this cell lineage and drawing these fate maps, then they formed the basis of the going into the next approach, which was the experimental biology. So there were a lot of methods by which the fate maps could be generated, and we will again talk about the detail of these generations and their usage when we talk about the experimental embryology as such. An important conclusion of cell lineage studies is that cells are constantly changing during embryology. So it's not that that the cells are giving rise to different cells, but they are constantly changing during the development phases which take place. The two main types of cells recognized by the early embryologists were the epithelial cells and the mesenchymal cells. The epithelial cells, which are tightly connected to one another in sheets or tubes, and mesenchymal cells, which are unconnected to one another and operate as independent units. They undergo morphogenesis through a limited repertoire of variation in cellular processes. The direction and number of cell division, cell shape changes, cell migration, cell growth, cell death, and changes in the composition of the cell membrane or secreted products. So basically, to answer the question of morphogenesis, the cellular level study was actually coming up, and they talked about the cellular basis of morphogenesis, and they tend to describe that, and therefore the generation of fate map formed the basis of studying this particular development. Thank you. So the generalization of Carl Ernst von Baer, which came to be known as von Baer laws, these generalization that Darwin built upon it and talked about the evolutionary embryology, which is again an anatomical approach to study the developmental biology. So Darwin interpreted von Baer's laws in the light of evolution and established a paradigm that relationships between groups can be discovered by finding common embryonic or larval forms. For example, the Nauplius larvae of a barnacle and a shrimp are very similar despite their radical divergence in later development. Darwin also noted that embryonic organisms sometimes make structures that are inappropriate for their adult form, but which show their relatedness to other animals. For example, the existence of eyes in the embryonic moles or pelvic rudiments in embryonic snakes. So one generalization led to the fact that all the embryonic structures are alike, and these relationship between groups can be observed at the embryonic level even if their adults are really different. But if you look at their embryos, they are very similar to each other and therefore lead to the classification of in fact different adults, different looking adults into the same phyla. Then he also talked about that the embryonic organisms sometimes have such structures in the embryonic region which is suited for the embryonic process. They might not need it in the adult stage, and that is not present, but at embryonic stages some of the structures which are required for the embryonic survival are also present. Darwin also argued that adaptations that depart from the type and allow an organism to survive in its particular environment develop late in the embryo. He noted that the differences between species within genera become greater as development persists. So again, deriving from the generalization of von Baer, that initially the more general characters are formed, and then the specialized structures are formed later during the development. So the adaptations, the adaptations to the environment for the survival usually comes later in the development, and therefore all the embryos are alike for different species. So in this evolutionary embryology, the two main things that came up to being was the presence of homologous and analogous organs. Homologous structures are those organs whose underlying similarity arise from their being derived from a common ancestral structure. For example, the wing of a bird and a forelimb of a human. However, analogous structures are those whose similarities comes from their performing a similar function rather than their arising from a common ancestor. For example, the wing of a butterfly and the wing of a bird both performs the function of flying, which therefore they are called as wing, but anatomically they are very different structures and they are not arising from the common ancestor. So evolutionary embryologists or evolutionary study aspect of the developmental biology lays emphasis on studying this homology and the analogous different structures. Now, one of the most celebrated cases of embryonic homology is that of the fish gill cartilage, the reptilian jaw and the mammalian middle ear. There is ample evidence that jaws are modified gill supports. First, both these sets of bones are made from neural crust cells. Most other bones come from the mesodermal tissue. Second, both the structures form from upper and lower bars that bend forward and are hinged in the middle. And third, the jaw musculature seems to be homologous to the original gill support musculature. So, although they are very different, the ancestry depicts there is a homology between the gill arches as well as the mammalian bone structure or the inner structure which is there. The upper portion of the second embryonic arch supporting the gill become the hyomandibular bone of the jawed fishes. This element supports the skull and links the jaw to the cranium. The hyomandibular bone happens to be near the otic capsule and bony material is excellent for transmitting the sound, because as mammals grow, now they have to hear in the medium which is called as air, which is very thin. So they need to develop a structure where the information or the sound can be transmitted in this lighter medium and therefore can be heard. So this hyomandibular, then while still functioning as a cranial brace, the hyomandibular bone of the first amphibians also began functioning as a signal transducer. So hyomandibular bone, which is actually the upper portion of the second embryonic arch supporting the gill in case of fishes, developed into the inner ear organ of the initial amphibians or the mammals. As the terrestrial vertebrates altered their locomotion, jaw structure and posture, the cranium became firmly attached to the rest of the skull and did not need the hyomandibular brace. The hyomandibular bone that seems to have become specialized into the stapes bone of the middle ear. So coming from the embryonic arches in the fishes, this hyomandibular bone actually developed into the stapes in the middle ear, because now it had to act as a sound transducer. So basically the homology, the ancestral origin is same, but in different species they are evolving. So these are the homologous structures which are there. So the middle ear bones of the mammal are homologous to the posterior lower jaw of the reptile and to the gill arches of the agnathan fishes. So evolutionary embryology lays emphasis on studying these homologous structures, because that tells us about how the evolution has taken place and how the development has altered in the light of this evolution, or vice versa, that evolution has taken place because the development has altered because of the needs of the environment that they found themselves in to be. So these things are related, and an evolutionary embryology studies that aspect. Coming to the next aspect in the anatomical approach is the medical embryology or the teratology. Now between 2 and 5% of human infants, they are born with a readily observable anatomical abnormality. These abnormalities may include missing limbs, missing or extra digits, cleft palate, eyes that lack certain parts, hearts that lack valves and so forth. Now the descriptive embryologists and evolutionary embryologists tend to study about why this is happening or how this is happening, but the more practical approach is taken up by the physicians. They became interested in embryos to know the cause of these birth defects in order to counsel parents as to the risk of having another malformed infant. In addition, the study of birth defects can also tell us how the human body is normally formed. Now, abnormalities caused by genetic events, for example, the gene mutation, the chromosomal aneuploidies and translocations, they are called as malformations. And the malformation, they often appear as syndrome, wherein the several abnormalities are seen concurrently. So, basically these abnormalities or medical embryology tends to study the defects into two categories. One is the genetic origin, which constitutes these malformations, and when many of them arises, then constitutes this syndrome. And another is what is called as the disruptions, which is basically the abnormalities due to exogenous agents, the certain chemicals or viruses, radiation or hypothermia, they are called as disruptions, and the agents responsible for these disruptions are called as teratogens. And the study of how environmental agents disrupt normal development is called as teratology. So, an important aspect of studying developmental biology is to get into the avenue of this medical embryology, where we talk about the different malformations, different syndromes, and try to find out the basis of the arising of these syndromes and malformations, as well as we study the teratology, that is the effect of certain exogenous agents that lead to these birth defects. The developmental biologists and clinical geneticists often study human syndromes and other diseases by studying animals that display the same syndrome, and these are called the animal models of the disease. So, ethically one doesn't start working on the human beings as such, but certain animal models are generated for different genetic defects and diseases, and developmental biologists, even the geneticists and the clinical developmental biologists, study on these animal models to know more about the genetic defects, or rather how the organism is normally formed. Then another aspect of studying the anatomical approach and studying developmental biology is to study it in the terms of mathematics. The goal of some investigators is to base the embryology on formal mathematical or physical principles. The pattern formation and growth are the two areas in which such mathematical modeling has given biologists interesting insights into some underlying laws of the animal development. Growth, in which the shape is preserved because all components grow at the same rate, is called as the isometric growth. The phenomena of the different growth rates of parts within the same organism is called as the allometric growth or allometry. For example, the allometric growth is seen in case of human beings in the picture. So, different pattern formations such as in snails and different growth patterns that are followed in the development of an individual. Mathematicians generally try to, or the physical people try to frame certain equations and study these kind of pattern generation which is there. For example, the formula for allometric growth for comparing money is invested at two different interested rates is y is equal to bx raised to the power of a by c, where a and c are the growth rates of two body parts and b is the value of y when x is equal to 1. So, if a by c is greater than 1, then that part of the body represented by a is growing faster than that part of the body represented by c. In logarithmic terms, which are much easier to graph, log y is equal to log b plus a by c log x. So, they study the different growth patterns by using mathematical equations and modeling the growth patterns onto it. So, that's again a different aspect of studying developmental biology in terms of equations. Then complex patterns may be self-generated by the mechanisms defined as what are called as reaction diffusion events, wherein the activator of a local phenomena stimulates the production of more of itself as well as the production of more diffusible inhibitor. So, this diagram basically represents the Turing model of reaction diffusion system, where he explains based on this mathematical modeling that how the complex patterns can develop. So, this is a system of pattern generation. The generation of periodic spatial heterogeneity can come about spontaneously when two reactants, for example, S and P, are mixed together under conditions that S inhibits P. P catalyzes productions of both S and P, and S diffuses faster than P. So, you can see in the first part of the diagram, that the S is rapidly diffusing and the P is slowly diffusing activator. So later on there is a higher peak of P that develops, and at where the peak of P is originating, the S peak is much lower because that is the inhibitor which is there. So initially there is a random concentration average of both S and P, but when P starts to develop, it stimulates the production of the S as well as stimulates the production of the P. Now because S is an inhibitor of P, this S inhibits the formation of more peaks around one of the peak that is developing, and therefore there is a consistent and spatial heterogeneity of the higher peak of P that exists, creating a kind of pattern. So such reaction diffusion systems described by Turing model describes the arrangement or the generation of the complex patterns during the development that exist. So these examples basically tells us that the development can also be studied mathematically by putting into the equations or by modeling and describing certain processes by modeling methods, and that also forms the aspect of the developmental biology studies. Now these were the approaches which were the anatomical approaches, and later on we will come to the experimental and genetic approaches. But before that we need to talk about the circle of life, that the stages of the animal development. One of the major triumphs of descriptive embryology was the idea of a generalizable life cycle. Each animal, whether an earthworm, an eagle, or a beagle, passes through the similar stages of development, and these stages of development are depicted in the picture. So the development starts when the germplasm or the when the oocyte and the sperm, they fuse together, or the mechanism of fertilization. So this is a generalized life cycle of all the organisms. So in all the organisms, the development starts from a single cell when there is a fusion of the gametes that take place. Particularly we are talking about in the sexual reproduction. Right now we are not talking about the asexual reproduction, that how the asexual reproduction existed and how it constituted, and then how the sexual reproduction came into being. Right now we are only talking about the generalization in the life cycles of the sexually reproducing organisms, that there are gametes that fuse together, and that process is called as the fertilization, and that is the first step in the developmental process. And the development starts from this single cell called as the zygote. This zygote then undergo mitotic divisions, a process which is termed as cleavage, to give rise to multicellularity. So single cell give rise to a multicellular organism by undergo different cleavage patterns. And different cleavage patterns, there can be a holoblastic cleavage, there can be meroblastic cleavage, and then in holoblastic cleavage there can be a different patterns of cleavages such as spiral cleavage, radial cleavage. We will talk about all these mechanisms in our later sessions. So basically the single cellular divides and give rise to the what is called as a multicellular ball of cells, which is called as the blastula in all the individuals. Now this blastula, these random ball of cells will now organize themselves, because there has to be the, we are seeking the answer to the question of morphogenesis, the ordered structure. So for the ordered structure to organize, the first step is the formation of the three germ layers, the ectoderm, endoderm and the mesoderm, the in-between mesoderm. So these germ layers occur by the movement or the cell migration after the cleavage is occurring, or basically simultaneously with the later cleavage stages. So this cleavage stages lead to the what is phenomena called as the gastrulation, and all the cells try to organize into three germ layers, the ectoderm, endoderm, so they move to the positions where they have to form the organ structure. So an ectoderm and mesoderm and the endoderm is formed during the process of gastrulation. And this gastrulation then leads to the formation of the organ. Now when the cells have reached the germ layers, have they reached their final position, then the tissues are arranged and the organs are formed, or the specialized structures are formed. So single cell gave rise to the multicellularity then gave rise to the ordered form of structure, and this ordered form and structure is now giving rise to the specialized organs. So they are forming the heart, they are forming the epidermis, they are forming the gonads, and all different kind of organs are formed during this organogenesis. Now, in some of the organisms, there are larval stages which are there, which then undergo different processes such as the metamorphosis to give rise to an adult. However, in certain organisms, this organogenesis is the formation of the adult. So, organogenesis occurs and then only the growth happens and the adult is formed. So, for example, in the case which is given here, which is the frog life cycle, there are larval stages which are there. There are different immature larval stages, and after this immature larval stages, the metamorphosis occur, or there are biochemical changes, structural changes, physical changes that occur that give rise to a sexually mature adult. Now, this sexually mature adult, by the process of gametogenesis, again produces gametes, and then this life cycle continues by the fusion of the gametes to give rise to the zygote by the process of fertilization. So, all the organisms have this kind of a circle of life or life cycle, and that is how the animal development proceeds through. Now, life cycle must also be adapted to the non-living environment and interwoven with other life cycles. So, these life cycle and the study of these life cycle is answering a question of differentiation, answering a question of morphogenesis, answering the question of growth, answering the question of reproduction. The question of evolution is identified by comparing the embryos of different individuals, or the specialized structures that develops during the evolution, and the environmental integration is very important. So, life cycle must be adapted to the environment in which they found itself, and therefore we seek to find the answer to the last question of the developmental biology, which is how the environment plays a role into the development. So, that is the different stages of animal development, and we will look at the animal development in detail for certain representative individuals during our talk about this developmental biology series. Now, looking at the developmental patterns among the metazoa. So, metazoans belong to one of the four major branches. Evolutionary, if we look at and the categorical divisions, if we look at, then there are different developmental patterns among the metazoa which is there. Now, metazoa belong to one of the four major branches: the sponges, the diploblasts, the protostomes, and the deuterostomes. So, if you look at this, sometimes the sponges are usually considered as a separate phyla, because they are usually considered as a separate phyla because they do not have the mesoderm. There are certain features that they are constituted under a separate class which is called as the parazoa, but generally we include them amongst metazoans because of certain features which are common to the other animal phyla also. So, they belong to, we will talk about these four branches. So, the sponges, also termed as the parazoans, they contain certain specialized cells which are called as the archeocytes. Now, these archeocytes are the specialized cells that can give rise to any cell of the body. So, they contain these specialized cells, archeocytes, which can give rise to any parts of the body. If a sponge is passed through a sieve, then the cells of that passes through the sieve tends to aggregate together. They tend to aggregate together, and then the archeocytes give rise to the different specialized cells which are formed. So, the sponges, they contain no mesoderm. As I said, they are diploblastic, or basically they contain only two layers. They have no true organ systems. Nor do they have digestive tube. They do not have circulatory system, nerves or muscles. They are very much unlike metazoans. So, some investigators tend to club these scientists into a separate line altogether, which is termed as the parazoans, but they do share many features such as gene regulatory proteins and a larval stage, suggesting that they share common origin with other animal phyla. So, some investigators club them with the other metazoans and talk about it as a different line, but some separate them and they tend to study it in the form of the parazoans only. So, sponges forms one of the major branches. Then came the diploblastic animals. Now, diploblastic animals are those who have ectoderm and endoderm, but no true mesoderm. These included traditionally the cnidarians, the jellyfish and the hydras, and the ctenophores or the comb jellies. Now, cnidarians and ctenophores constitute the radiata, revealing that they have got a radial symmetry like that of a tube or a wheel. In these animals, the mesoderm is rudimentary, consisting of sparsely scattered cells in a gelatinous matrix. So when we look at the evolutionary patterns of metazoans and we keep aside sponges, then there are certain cnidarians and ctenophores which have a radial symmetry, which show radial symmetry, and they have only two germ layers which are there. They do not have a true mesoderm which is formed. So these are termed as the diploblastic animals as against the triploblastic animals which have got 3 germ layers and usually comes under bilateria because they are bilaterally symmetrical. So most metazoans have bilateral symmetry and 3 germ layers. That's considered to be the triploblastic animals. The evolution of the mesoderm enabled greater mobility and larger bodies because it became the animal's musculature and the circulatory system. So the triploblastic animals developed the 3rd germ layer and therefore they have much more flexible and larger organisms than what the diploblastic animals were. So there were sponges as the one branch, the diploblastic animals as the other branch, and the triploblastic animals as the 3rd branch. Now triploblastic animals or animals of the bilateria are further classified as protostomes or deuterostomes. So stome is basically the mouth, the protostomes or the deuterostomes. So protostomes basically means the mouth first. They include the mollusk, the arthropod and the worm phyla, and they are so called because the mouth is formed first at or near the opening to the gut, which is produced during gastrulation. The anus forms later at another location. The coelom or the body cavity of these animals forms from hollowing out of previously solid cord of the mesodermal cell. So protostomes form another branch of the metazoans where the mouth comes first and anus develop from a different regions which is later on. The mollusks, arthropod and the worm phyla are basically the protostomes. There are 2 major branches of the protostome. One is termed as the ecdysozoa, includes those animals that molt. Its major constituent is Arthropoda, a phylum containing insects, arachnids, mites, crustaceans and millipedes. So basically the insecta phyla comes into this ecdysozoa because they leave their external cuticle, they molt and then the next larva or the adult emerges out of it. So they shed their cuticle periodically and therefore they are termed as ecdysozoa. The second major group of protostomes are the Lophotrochozoa. They are characterized by a common type of cleavage which is termed as a spiral cleavage, a common larval form and a distinctive feeding apparatus. These phyla include the annelid, mollusks and flatworms. So all these comes into the protostomes because the mouth develop first and the anus develop at a later stage, but depending upon whether they molt or whether they undergo a specific type of cleavage, they are categorized into 2 further subdivisions. Now phyla in the deuterostomes in lineage include the chordates and the echinoderms. Now first in deuterostomes, the mouth opening is formed after the anal opening. Also, whereas protostomes generally form their body cavities by hollowing out a solid mesodermal block called as the schizocilous mechanism of the formation of body cavity, most deuterostomes form their body cavities from mesodermal pouches extending from the gut, and this is termed as the enterocilous formation of the body cavity. So deuterostomes not only differ from the development of the mouth or the anus which comes first, but also differs in the way their gut cavity develops. The evolution of organisms depend on inherited changes in their development, and one of the greatest evolutionary advances was the formation of the amniote egg that occurred during among the deuterostomes. The amniote egg allowed the vertebrates to roam on land far from the existing ponds. The amniote egg carries its own water bodies and food supplies. It is fertilized internally and contains yolk to nourish the developing embryo. Moreover, the amniote egg contains 4 sacs, the yolk sac, the amnion, the allantois and chorion, which have different functions which are there. Then this entire egg is encased in a shell that allows the diffusion of oxygen but is hard enough to protect the embryo from environmental assaults and dehydration. So we talked about the evolutionary patterns of metazoa, that how they diversify into 4 different branches and gave rise to the different individuals, and that differentiation is linked to the development that they are undergoing. So sponges have a different kind of a germ layer arrangement, diploblastic animals, triploblastic animals, then within triploblastic elements we have different kind of a different categories based again upon the development that occur. So this entire aspects constitute the anatomical approach in studying the developmental biology, and in later session we will talk about the experimental embryology and genetic embryology, that how this genetic aspects approaches are there, or what experimental approaches are studied for the developmental biology as such.
Dear friends, for the paucity of time, we have to stop our lecture here. On that note, we would like to thank Dr. Dogra for coming to our show and delivering this wonderful lecture. And thank you, dear friends, for watching our show. Stay tuned and keep watching. Thank you.